Hello everyone, welcome back to my YouTube channel, Life Sciences by Tumi Silani. We are fresh from lesson 2 on our lesson series on meiosis. This is lesson 3. We will be discussing genetic variation. The background for this lesson is based on lesson 2 on the previous video. So please, for you to be able to understand the content in this video, it is very much important to watch the second lesson in the second video where we explain the process of meiosis. In the meantime, I would appreciate it if you like, comment, subscribe, and share the link to this video. My name is Tumi Silani. I am a teacher at Milner High School here in the Northwest Province. Like I said, this will be lesson 3 on the lesson series of meiosis. I hope that you will enjoy this lesson. First of all, let us break down the different lessons of this lesson series so that we can keep track of what we have done so far and what we are going to do in this video and in the next video. So, I have decided to divide the lesson series on meiosis into 5 lessons. Each lesson is contained within its own individual video. So far, we have done lesson 1, where we looked at the introduction to meiosis. This is all the preliminary concepts that you need to learn and understand for you to be able to comprehend fully the process of meiosis. On the previous lesson, lesson 2, we looked at occurrences that happen during meiosis 1 and meiosis 2. On today's lesson, we will be building up from lesson 2, where we will be looking at genetic variation and how meiosis ensures genetic variation. So like I said, it is very much important for you to have background knowledge on lesson 2 before you even look at the video on lesson 3, because this lesson is a build up from lesson 2. Okay, so there are 2 ways in which meiosis ensures genetic variation. One would be crossing over, as well as random arrangement of chromosomes. Okay, so that is what we will be describing today. And the 4th and final lesson will be on abnormal meiosis. There we will be looking at non-disjunction. Let's get right into the lesson. Okay guys, now we cannot speak about how meiosis ensures genetic variation without speaking about what genetic variation actually is. Okay, so on this slide, I will be trying to explain to you what genetic variation is. So, the word variation quite simply just means difference. Okay, so among the species of different organisms, there is difference. Let's look at human beings, for example. Human beings, we are the same species. However, we show difference. Okay? So there are 2 types of difference that are there, or 2 types of variation that is there in nature. Firstly, there is physical difference or physical variation. This is scientifically known as phenotypic variation, but let's just call it physical difference for now, or physical variation. Like I said, even among humans belonging to the same species, we differ. We have people of different skin tones, people of different hair textures, hair colors, and etc., and etc., and etc. Let me bring it back home. Even within siblings of the same parents, siblings of the same mother, and siblings of the same father in human beings, we might be similar. You might be similar to your brother, you might be similar to your sister, but you are not 100% identical. Between you and your siblings, you show physical variation or physical difference. Okay, now let's check this picture to make a note. So here we have two absolutely adorable puppies. They are puppies of the same litter, meaning they were born at the same time and were bred by the same mother and the same father at the same time, and they were born at the same time. Okay, now when you look at them physically, they do not look alike, even though they were born at the same time from the same set of parents. Okay, they have so many differences amongst them. This type of difference, the difference in the physical body, is called physical variation or phenotypic variation. Okay, it is the difference that we are able to see in the bodies or on the bodies of these particular puppies. But this begs the question: they have the same parents, they were bred at the same time, and they were born at the same time. How are they physically different from each other? Why do they show this physical variation? Now, how we look depends on our genetic variation. Okay, so the physical difference is because of the genetic variation. That is the subject of today's lesson. So the physical variation or the phenotypic variation is not for today. What we are focusing on today is the genetic variation. What is genetic variation? Genetic variation is having a different combination of genes in the DNA. What did we say a gene is from lesson 1 of this lesson series? We said that a gene is a small segment or a small piece of DNA that codes for particular characteristics. Okay, so a gene, we might as a species have the same type of genes, but the way that those genes are composed and the way that they are expressed in the physical body is not the same. So the traits or the characteristics that we have, for example, depend on the composition of genes and how those genes are expressed in our physical bodies. So there is, for example, a gene for eye color. Okay, on you it might code for a hazel color, and then on your sibling that particular gene might code for a darker brown color. We have a gene for another characteristic, let's say for example skin tone. It might express being light skinned, or it might express being more darker skinned. We have a gene for foot size, for example. One might have a larger foot and the other person might have a smaller foot. We have a gene for the shape of the nose. One person might have a slightly bigger nose. Another person might have a slightly smaller nose. So our physical characteristics depend on how our genes are composed. Okay? So different combinations of genes will express different variations of physical features. Now, the reason why we are physically different is because our genes are composed differently, and they will express themselves in different variations. So our physical variation is dependent on our genetic variation, on how our genes are composed in our DNA. Okay, guys? Now, where does this all start? Why do we have different combination of genes, or why do we show genetic variation? Meiosis. It starts from meiosis. I will use the following picture to illustrate this. Remember that meiosis from lesson 2 on our lesson series on meiosis, we discussed that we would have an original mother cell that will undergo meiosis 1 and meiosis 2. And then at the end of meiosis 2, we will have 4 daughter cells. And these 4 daughter cells would be gametes. So if these were sperm cells, let's say, for example, these were 4 sperm cells, okay, that result from meiosis. Let's say, for example, these are 4 sperm cells that result from meiosis in males. When you check each individual sperm cell there, all of them have a different combination of colors, okay? The chromosomes have different combinations of colors. Now, these combinations of colors represent the genetic material. It represents the genes within the chromosomes. So when you look at all of them, they show a different combination of genes. Therefore, we say each individual sperm cell is different from each other as it results from meiosis. Remember, we said that we have 3 functions or significances of meiosis. Number 1, meiosis produces gametes. So in this instance, these are 4 sperm cells, the male gametes. Secondly, it ensures that the gametes are haploid. They contain only one set of chromosomes. And then the last function, we said that meiosis ensures genetic variation, or in other words, that each gamete that is produced by meiosis is genetically different from each other. Okay? So as you can see over there, each sperm cell has a different combination of chromosomes with a different combination of genes. Remember what we said? We said that our physical bodies or our physical difference depends on how our genes are composed and how our genes will be expressed in our physical bodies. All right? Now, for instance, if this sperm cell were to fertilize an ovum, the genes within that offspring will be composed in a certain way, as opposed to when this sperm cell fertilizes a different ovum. We will have a different combination of genes in that child, and that child therefore will express those genes in a different way. So this explains exactly why we show physical genetic variation. We show physical genetic variation because the gametes that fertilize to make us have a different combination of chromosomes with a different combination of genes. Therefore, if we have a different combination of genes, the way in which our genes are expressed to make our physical bodies will also be different, even though we come from the same parents in the case of siblings. Okay, guys? So, the subject of today's lesson is to explain how meiosis is able to produce gametes that are genetically different from each other. Okay, so meiosis does this in 2 ways. We are going to study both those 2 ways. Remember, guys, we said that there are 2 ways in which meiosis ensures genetic variation. So the first way is crossing over during prophase 1. So we will be starting with crossing over during prophase 1. And then later on, we will move to a different way in which meiosis ensures genetic variation. Okay, now for the first question. What is crossing over? What is it that we are talking about when you're talking about crossing over? So this is a description of the term crossing over. Crossing over is the exchange of genetic material. Is the exchange of genetic material. If you don't want to say exchange of genetic material, you can say exchange of DNA, because DNA is genetic material of the cell, between chromatids of homologous chromosomes. Okay, so the exchange of genetic material, or the exchange of DNA between chromatids of homologous chromosomes. Guys, please be particular about the definition of this term. Okay, what is being exchanged is the genetic material, not the genes. Okay, it is the DNA, portion of the DNA that is being exchanged. Do not say genes, because you will be discredited. It is the exchange of genetic material. We are exchanging genetic material, or DNA, between chromatids, please, of homologous chromosomes. Okay, so the genetic material is being exchanged on the chromatids of the homologous chromosomes. Question number 2. How does crossing over lead to genetic variation in gametes? How does crossing over lead to genetic variation in gametes? So what this question is asking is that, how does crossing over lead to the gametes being different from each other as they result from meiosis? Okay, so this is how you are going to answer this question, and I will show you on the following slide how this happens. So the answers are: during prophase 1, for 1 mark, chromatids of homologous chromosomes overlap, 1 mark. There will be exchange of genetic material, guys, not genes, please. There will be an exchange of genetic material, or there will be an exchange of DNA at the chiasma. At the chiasma, this is a 2 mark statement. I will explain what a chiasma is, such that each chromosome of the homologous pair will have a different combination of genetic material. Okay, so this is an explanation on how genetic variation occurs. Remember, on this answer, we were giving a definition of what crossing over is, but here we are describing how crossing over leads to genetic variation in gametes. Okay, so after meiosis, each gamete will receive a different combination of chromosomes with different combinations of genetic material. So during prophase 1, chromatids of homologous chromosomes overlap. There will be an exchange of genetic material at the chiasma, such that each chromosome of the homologous pair will have a different combination of genetic material. After meiosis, each gamete will receive different combinations of chromosomes with different combinations of genes. No, not genes, genetic material. Okay, now let us check how that happens exactly. Okay, now in this slide, I will be showing you how crossing over takes place. Now, we have step number 1. I'm going to be showing you a homologous pair of chromosomes before crossing over. There we go. Now, remember, what did we say a homologous pair of chromosomes is? A homologous pair of chromosomes are a pair of chromosomes. One is paternal. We get it from the sperm of the father, and one is maternal. Get it from the ovum of the mother. Okay, they are the same size. They carry the same genes at the same locus, if you can still remember from lesson 1. Okay, so this is a homologous pair of chromosomes. They have different colors representing different genes. So this one is all purple, and then this one is all gray. Excuse me, I'm colorblind, so if this is not purple, please don't make fun of me. Okay, whatever color this is, I'll just call it purple. But I know this is green. I can see that very well. All right. Now, we have the maternal and paternal chromosomes with the maternal and paternal genetic material. Not genes, genetic material. Okay. Now, step number 2. We said what happens. We said that chromatids of homologous chromosomes are going to overlap. Please do not say that they touch. Okay? Use the word overlap. Do not use the word touch. So there we have chromatids of homologous chromosomes. They overlap. Okay? Then there will be an exchange of genetic material or an exchange of DNA at the chiasma. What is a chiasma? A chiasma is the point where these two chromatids of homologous chromosomes overlap. So that point where they overlap and genetic material is being exchanged, that is what we call the chiasma. So where they overlap, that's only where the genetic material will be exchanged. What do we mean by exchange? And please do not use the word they share DNA or they share genetic material. Please use the word exchange. By exchange, what do we mean? We mean some of the purple genetic material from the maternal chromosome will go to the paternal chromosome, and some of the genetic material from the paternal chromosome will go to the maternal chromosome. Okay, but not everywhere, only at the chiasma, only at the point where they overlap. One chromosome exchanges genes with the other chromosome. Some of the genetic material from this chromatid goes to that chromatid, and some of the genetic material from that chromatid moves to that chromatid. Okay, they don't share genetic material, they exchange genetic material. Okay, now there is only one point of overlap there. Okay, they overlap at one point, but it is not always that they overlap at one point. They might overlap at 2 points, the bottom parts of the chromatids and the top parts of the chromatids as well. Now, it means that we would have 2 points of exchange of genetic material. When that happens, we say that we have 2 points of exchange of genetic material, and then we will say we have 2 chiasmata. Okay, the plural for chiasma is chiasmata. Okay, not chiasmas, chiasmata. So singular, 1 chiasma and many chiasmata. Okay, so after they exchange the genetic material at the chiasma, what are they going to look like? Step number 3, after crossing over. This was before crossing over. This was during crossing over. Now, what are they going to look like after crossing over? Right. The maternal chromosome is going to look like this. Now, what happened to it? The paternal chromosome will look like that. Now they look different all of a sudden. What happened to it? Now, the genetic material that is represented by gray is the genetic material from the paternal chromosome. It was exchanged from the paternal chromosome and it went to the maternal chromosome. Okay? When you look at the paternal chromosome, you will see that the bottom left chromatid is now purple. Why is it purple? Some of the genetic material from the maternal chromosome went to the paternal chromosome. So there was an exchange of genetic material at the chiasma. You may be asked to draw chromosomes as they would appear in the cell after crossing over. Okay, you will be given a diagram. For example, you might be given this diagram. Okay, one might be shaded black and then the other one will be left as white. Please, when you draw chromosomes after crossing over, check where the chromatids overlapped. You will only exchange the colors or the numbers if they give you numbers where they overlapped. It is not the whole chromosome that is going to overlap or the whole chromatid that is going to overlap. Okay, guys? Only where they overlap. That is where there is going to be an exchange of genetic material. Hence, these chromosomes show crossing over at the bottom inner chromatids, not the whole chromosome overlapped. Check, the left chromatid of the maternal chromosome did not overlap. Hence, the left chromatid kept its color. Okay? The top part of the right chromatid did not overlap. Hence, it is still purple. But the bottom right chromatid overlapped. So, hence, it is carrying some of the paternal genetic material. Let's look at the paternal chromosome. The whole right chromatid did not overlap. Hence, it maintains its gray color. The top part of the left chromatid did not overlap. Hence, it is still showing gray. But the bottom left chromatid, as you can see over there, overlap. So it took some of the genetic material from the maternal chromosome. Hence, it is purple. Okay, so at this stage, each chromosome of the homologous pair has a different combination of genes, or rather a different combination of genetic material, as you can see there, or we can say a different combination of DNA. All right. So that is crossing over during prophase 1. What you need to know is what is crossing over, the definition. We said that the definition of crossing over is when chromatids of homologous chromosomes exchange genetic material during prophase 1. And of course, it occurs during prophase 1. And then you need to also be able to describe how crossing over leads to genetic variation in gametes. Okay? And the answer to that, we said that chromatids of homologous chromosomes will overlap. There will be an exchange of genetic material or exchange of DNA at the chiasma. Okay? Now, each gamete after meiosis will receive a different combination of chromosomes with different combinations of genetic material. Okay, with that being said, let us look at the second way in which meiosis causes genetic variation. Remember, guys, we are still on the topic or the subtopic of genetic variation. We have just seen how crossing over that takes place during prophase 1 causes genetic variation, or causes that each daughter cell of meiosis becomes genetically different from each other. That is not the only way to ensure genetic variation in terms of meiosis. The second way in which meiosis ensures genetic variation is random arrangement of chromosomes in metaphase 1. So let us describe how that further ensures genetic variation in the gametes that are produced by meiosis. Okay, guys, now here we are describing how random arrangement of chromosomes at the equator of the cell during metaphase 1 or metaphase 2 leads to genetic variation. Just a reminder from lesson 2, we said that during metaphase 1, if you can still remember, homologous chromosomes will align at the equator of the cell in pairs. Okay? Now, after that, the centrioles will produce spindle fibers, and these spindle fibers will attach themselves to the centromere of the homologous pair of chromosomes, such as that. Okay? And during metaphase 2, we said that individual chromosomes will line at the equator of the cell. Remember, equator just means the middle of the cell. Okay? And then the centrioles produce spindle fibers, okay, which will attach themselves to the centromeres of the individual chromosomes, such as that. There we go. Remember. Now, let's say for example, this is chromosome pair 1, and this is chromosome pair 2. Okay? This will be individual chromosome 1, and this will be individual chromosome 2. Okay? So, they do not necessarily pair themselves in a specific order. Remember, at the equator of the cell, if this was a human cell, we would have 23 pairs of chromosomes that are lining themselves at the equator, 23 pairs. They do not necessarily align themselves in pair 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 until pair number 23. In one cell, they might align themselves like that. In another cell, the first pair might be pair, for instance, might be pair 3 as the first pair, and the second pair might be pair 19, and the third pair might be pair 23, the fourth pair might be pair 2. Okay? On another cell, it might be a different order. In the different cell, it's a different order. So that is what we mean by they pair themselves randomly. By randomly, we mean that they don't pair themselves in any particular order. In each cell that is dividing, they align themselves in different orders. Therefore, they pair themselves randomly. Why is this important? I'm going to show you in this slide that the random arrangement of chromosomes in pairs in metaphase 1 and individually in metaphase 2 would actually warrant the cells being pulled to opposite poles of the cell in random combinations, in different combinations, and at the end of meiosis, each daughter cell will receive different combinations of different combination of chromosomes, which have different combinations of genetic material. Okay, now let us contextualize this. Let me just erase and make space for the text. Bear with me. Okay, there we go. So first question, how does random arrangement of chromosomes in metaphase 1 lead to genetic variation? Okay, so the answer is, remember you need to know this answer. The answer is, during metaphase 1, you get a mark for indicating which phase. homologous pairs of chromosomes align at the equator of the cell randomly. Okay? And then they will be pulled to opposite poles of the cell in different combinations during anaphase 1. At the end of meiosis, each gamete will receive different combinations of chromosomes with different combinations of genetic material or with different combinations of DNA. Okay, now remember we said that genetic variation starts from the sperm cells and the ova that are produced by meiosis. Okay, males produce millions of sperm cells each and every single day. All those millions of sperm cells are genetically different from each other. Why do we say they're genetically different from each other? Because they contain different combinations of chromosomes that have different combinations of genetic material or different combinations of DNA. Where did it all start? During metaphase 1, homologous pairs of chromosomes align at the equator of the cell randomly. They will be pulled to the opposite poles of the cell in different combinations during anaphase 1. And then at the end of meiosis, there will be different combinations of chromosomes containing different combinations of genetic material. Okay. Now, if you want to describe random arrangement in terms of metaphase 2, if you want to describe random arrangement of chromosomes in terms of metaphase 2, you are welcome to do that. But remember, we will be replacing the word metaphase 1 with metaphase 2. We will be replacing homologous pairs of chromosomes with individual chromosomes. And then we will be replacing anaphase 1 with anaphase 2. Okay, let us check. During metaphase 2, individual chromosomes align at the equator of the cell randomly. Chromatids or unreplicated chromosomes will be pulled to the opposite poles of the cell in different combinations during anaphase 2. At the end of meiosis, each gamete will receive different combinations of chromosomes with different combinations of genetic material. All right? Thus, each gamete will be genetically different from each other. Okay, so this is an explanation. As you can see, it is a good, expensive 6 marks. So if I were you, I would be taking notes. And as you are taking notes, please include these ticks so that you know how markers may mark this question. Okay, now in this slide, I will be showing you what we talked about on the previous slide. Okay, let's check. Chromosomes may arrange in any order during metaphase 1 or metaphase 2. That's what we mean by random arrangement. The word random means in any order, okay? In any order, not the same order, in any order. Take, for instance, possibility 1. In this cell, we have this combination. This is homologous pair 1 and this is homologous pair 2. They have aligned themselves at the equator of the cell in this combination. Okay, let's say, for example, let's give these chromosomes names. Let's say this is chromosome A, this is chromosome B, this is chromosome C, and this is chromosome D. Excuse me. Let's say this is chromosome D. Now, please look at the chromosomes. Look at the chromosomes and how they are arranged in this cell. So this is pair, chromosome pair 1, and this is homologous pair 2. Okay. In another cell that is dividing, they will be arranging in a different combination. Check with me. There we go. Now, this is a different arrangement there. When you check in this chromosome, chromosome pair 1 is at the bottom, and then chromosome pair 2 is at the top. So the way that they arrange themselves is random, it's different from each other. And what else should we note? This is chromosome number 1. Look at where it is on the cell. And this is, chromosome A. This is chromosome A. Look at where it is on the cell. And this is chromosome B. Okay? Now let's check chromosome C. Chromosome C is there. And chromosome D is there. So you can see that the way in which chromosomes line themselves at the equator during metaphase 1 is different in the different cells that are dividing. Okay, so what will this result in? Remember, at the end of meiosis 1, may I just erase? At the end of meiosis 1, the cell is going to divide in half. Okay? So, on this cell, these chromosomes will be pulled to that pole of the cell. Those two chromosomes will be pulled to that pole of the cell during anaphase 1. And then when the cell divides at telophase 2, we are going to have that chromosome in this daughter cell, as well as that chromosome in that daughter cell. On a different daughter cell, we will have that chromosome on the daughter cell, and then we will also have that chromosome on that daughter cell. Okay, now let's look at this cell. Okay, let's look at this cell. On this cell over here, because they align themselves at the equator of the cell in a different combination at the equator of the cell, we expect that the daughter cells will contain a different combination of genes as compared to the daughter cells of this cell over there. So when the cell divides like that, that daughter cell will have that combination of genes. Now look at these two chromosomes. Look at these two chromosomes and look at the combination of chromosomes in this daughter cell. It is not the same as that one. It is definitely not the same as that one. Okay, this chromosome, there it is, but that one is not the same as that one. That one is majority purple and minority gray. That one is majority gray, and minority purple. It's not the same chromosome, okay? And then on the second daughter cell, we are going to have that chromosome as well as that chromosome. When you check, this daughter cell has a different combination of chromosomes with a different combination of genes. Remember, why are they different colors? They are different colors because of crossing over that took place at prophase 1. Now, when you check them, when you check this combination of chromosomes in this daughter cell, it's not the same as there. It is not the same as there, and also not the same as there. Why are the are the combination of chromosomes in each daughter cell different? They are different because the way in which the homologous chromosomes line themselves at the equator was at different combinations. So they will be pulled to the opposite poles of the cell in different combinations, and then the daughter cell will also contain different combinations of different combination of chromosomes with different combinations of genetic material. So this is after meiosis 1. And after meiosis 2, remember what's going to happen. The cell is going to divide like that, okay? So when it divides like that, remember the the chromosome is going to split, and on that chromosome, you are going to receive that unreplicated chromosome as well as that unreplicated chromosome. And then at the second daughter cell, we will have that unreplicated chromosome and that unreplicated chromosome. And then when this cell divides under meiosis 2 as well, it will divide in half, and then you are going to have that unreplicated chromosome as well as that unreplicated chromosome. And same goes, that unreplicated chromosome as well as that unreplicated chromosome over there. Okay, so in these two daughter cells, when they divide via meiosis 2, the same is going to happen. The same is going to happen. We are going to have different combinations of chromosomes with different combinations of genes. So when you analyze all these daughter cells, all of them have different combinations of chromosomes containing different combinations of genetic material. Where did it all start? It all started because the way in which they aligned themselves, the way in which the chromosomes aligned themselves in metaphase 1 and in metaphase 2 was random. Okay, guys? So if these were sperm cells, if all of them were sperm cells, remember I said to you, meiosis produces millions of sperm cells every single day in males. So if all of these were sperm cells, for instance, all these sperm cells would be genetically different from each other. So during sexual reproduction, if all of these sperm cells fertilized different ova, the children or the offspring resulting from these 8 fertilizations will be genetically different from each other. Why? Because the sperm cells and the ova that made that offspring contain a different combination of genes, with a different combination of chromosomes containing a different combination of genes. So they show genetic variation. That's why dogs having a litter of, let's say, 6 puppies at the same time, it means that it is 6 different sperm cells that contain 6 different combinations of genetic material, 6 different combinations of chromosomes. They fertilized 6 ova containing different combinations of genetic material and different combinations of chromosomes. Okay, that's why the offspring will be genetically different as well. All right, I hope this makes sense. Now, I am going to fit in the differences between meiosis 1 and meiosis 2 into this lesson, and then we are going to move on to exam type questions. So guys, on the next slide, I have included the differences between meiosis 1 as well as meiosis 2. You need to know the differences between phases of meiosis 1 and phases of meiosis 2. For instance, prophase 1, how does it differ from prophase 2? Metaphase 1, how does that differ from metaphase 2? Anaphase 1, how that differs from anaphase 2? And then telophase 1, How does that differ from telophase 2? You need to know the key differences between those phases, because you might be asked to tabulate them, or you might be given a diagram and for you to identify a phase as well as explain why you say that particular phase is. So there's a certain way that you need to describe these phases. Okay, let's get right into it. For instance, they ask in this format to tabulate the differences between meiosis 1 and meiosis 2. The instruction says tabulate. The word tabulate means put in a table. You have to put in a table. Those differences must be put in a table. Now, it is easier for me, I don't know for you, it is easier for me to look at the differences per phase, okay? Per phase. Prophase 1, prophase 2. Metaphase 1, metaphase 2, and so on, for you to maximize the chances of getting correct marks. So, let's look at during prophase. Prophase 1, how does it differ from prophase 2? Chromosomes are arranged in homologous pairs in the cell during prophase 1, 1 mark. But in prophase 2, the chromosomes are arranged individually in the cell. They do not form homologous pairs. 2nd mark. In prophase 1, there is crossing over that takes place. However, in prophase 2, there is no crossing over that takes place. We move to the metaphases. In metaphase 1, chromosomes align in the equator of the cell in homologous pairs. However, in metaphase 2, chromosomes align in the equator of the cell individually. They are aligned individually or singly. They do not align at the equator of the cell in homologous pairs. In the anaphases, do we have a difference? Yes, we do. In anaphase 1, as the spindle fiber contracts, whole chromosomes are pulled to the opposite poles of the cell. However, in anaphase 2, chromatids are pulled to the opposite poles of the cell, or unreplicated chromosomes. When you say unreplicated chromosomes, that will also suffice. Unreplicated chromosomes or chromatids are pulled to the opposite poles of the cell. Remember that replicated chromosome splits, and chromatids or unreplicated chromosomes are pulled to opposite poles of the cell. Telophase 1, telophase 2. We have two differences. 2 daughter cells are produced in telophase 1, but in telophase 2, we have 4 daughter cells that are produced. Right. At the end of telophase 2, daughter cells contain replicated chromosomes. So all the daughter cells have chromosomes that are, or that have 2 chromatids each, but at the end of telophase 2, unreplicated chromosomes result in the daughter cells, meaning all the daughter cells, all 4 of them, will contain chromosomes with 1 chromatid each. Now, this difference is random. It does not fall under any phase in particular. Generally, after meiosis 1 reduces the number of chromosomes from diploid to haploid. But remember what we said when we were looking at the different phases of meiosis. We said that there is no reduction of chromosomes after meiosis 2. So, in the exam, the examiners are not going to ask you to tabulate differences for 15 marks. Absolutely not. that is way too many marks for one question. So they might ask you to tabulate 3, tabulate 4, give you maximum marks of about 6, which will be 7. Why 7? When we count, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 marks. However, you receive 1 mark. I don't want to say free mark, but it's a free mark, okay? Well, it's not free, but you receive 1 mark for following the instruction, putting the differences in a table. So you'll see the markers usually will write T next to the table and make a tick like that. This T is for the drawing the table. But drawing the table alone would not qualify you for this 1 mark. The headings of the columns of your table are the ones that give you 1 mark. The instruction says tabulate 2 differences between meiosis 1 and meiosis 2. Excuse me, there was a mistake there as well. It must be meiosis 2 there. All right? So you must write as a heading for the first column either meiosis 1 or meiosis 2, doesn't matter. And this side, you must write again meiosis 1 or meiosis 2. When you've written meiosis 1 this side, you write meiosis 2 that side. When you've written meiosis 1 that side, you write meiosis 2 that side. So the headings of the columns of your table are the ones that qualify you this mark. Okay, so if you just draw the table without writing the correct variables or the correct headings of the columns, you will not get this 1 free mark. All right, and then the content will constitute the rest of the mark allocation. Okay.